Thank you so much. And apologies in advance for that mouthful of a title, but trying to condense down a few years' worth of work was a little bit tricky. But so, like Xander said, I'm going to be talking about understanding cells in context. So looking at quantitative methods for assessing what cells are doing and what that means for mammalian development. And so today, what I'm first of all going to start with is my disclaimer. So that should perhaps be interpreted as a warning for this talk. But then I'll give you the briefest of introductions to mammalian development and the section of that I'm interested in. And then I'm going to be talking about a couple of these quantitative approaches that I developed during my PhD. And these include quantifying static cell neighbourhood. This is quite a simple approach. But then moving on and spending a bit more time on how I'm starting to look at what's happened to cells in the past and how that impacts what they then eventually do and become. And then I'm going to just try and take a step back because you may be a little bit bored of mammalian development by that point. And so just to talk about and show you how general some of these approaches can be and what we can use them for besides looking at mammalian development. So, starting off with my disclaimer, I can't choose a single topic and never have been able to. So if for some reason you were all looking at my CV, you would see that I never pick one subject. So started off, I was trained as a medical physicist, and then, like Xander said, I started my PhD. I was really fortunate to get a Wellcome Trust-funded PhD position where I got to play around in the lab, but then also develop some of the more theoretical elements on the side. So I've got lots of random biology facts to tell you about, but I'll try and keep them to a minimum. And then I did my first postdoctoral position at the University of Manchester, applying some of those concepts I developed in my PhD to something completely different, but still within biology. And then, just in case anyone was interested, I've taken a bit of a career shift now. So I'm a statistician, hopefully, but what I work on is reviewing technology appraisals which are submitted to the National Institute for Health and Care Excellence from pharmaceutical companies, so looking at the cost effectiveness of new drugs and so forth. So really different. I'm not going to be talking about that today, but perhaps in coffee, if you want to talk about it, I'd be more than happy to. Okay. So for the purpose of this talk, most of the work that I'm going to be talking about was actually done within my PhD. It's a bit of a slow burner. These things take a bit of time to get your head round, and so that's why I'm only talking about it now. But there are links later on within my research. So to start off with the big picture, and I think in applied maths we're really bad. We sometimes forget about the big picture. Why are we doing this? Why are we asking for this funding and so forth? But what I'm going to be talking about is early mammalian development, and what that relates to is the early embryo. And so this can be quite a controversial subject, so it's important to understand why we're doing it. So in vitro fertilization was first successfully performed back in 1978, but the success rates still remain quite low, and this can be due to lack of our knowledge about what the culture conditions of the embryos should be. How do the embryos like to develop? What kind of environment? Do they like it quite warm? Do they like it quite cold? I'm talking about more sophisticated things than that, but it's the basic concept. But also, our knowledge of what a good or naturally developed embryo looks like is still not fully formed. We don't know how to say, yes, this is a really good embryo. We think this will result in a successful pregnancy, versus one which we think may have some difficulties in developing, especially when we're culturing them outside of the body. Another thing, a lot of my research here, I'm going to be talking about the microenvironment, so what is going on around cells in their neighbourhood. And this is really important within cancer progression. So the way that cancers metastasize, and if we can understand that better, then we can maybe develop better treatments and bespoke therapies to help treat cancer in a variety of settings. And then finally, this is the most non-biology explanation, but they've got cool properties, and it lends itself to regenerative medicine approaches. And so this all kind of fits in here. There's a lot of stuff going on if we can start to understand what's actually happening. Okay. So because I can't help myself, and I'll be showing lots of images throughout this, I'm just going to give a crash course of the stage in development that I'm interested in. And so We call this the pre-implantation period. So what this refers to is the point between when the egg is fertilised by the sperm and the point at which, all the way up this end, we have the development of this gorgeous structure here with some blue cells on the outside, yellow cells on the inside, and then a border of these orange cells with a big empty space, this cavity. And this is really important, that during this process we have the specification of these 3 cell types, so the 3 cell colours, and that they're organised in the correct way. If they're not, then the embryo can't implant, and ultimately the pregnancy will not continue. So it's a really important stage within our development. And what I'm showing you here is the pre-implantation period in the mouse, but it's very much mirrored in the human. It's slightly different timings and slightly different numbers of cells, but otherwise it's largely preserved. And so this serves as a really good model system. Now, I'm calling them blue cells, yellow cells, and orange cells. I'm going to try my best not to slip into the biological words because it just gets too much. But I will mention them here. Don't worry about them too much later on. But we've got these 3 cell types. So the trophectoderm, which are the blue ones on the outside, the yellow cells, which are the epiblast cells. These are the ones which actually go on to form all of the other cells in your body. So these are those ones linking to regenerative medicine that I was just speaking about. And then we've got the primitive endoderm cells here, which border that cavity. And what's really important at this stage is this is actually a 3-dimensional structure. So I've just shown you a nice 2D schematic. It's all very nice and tidy. But this is actually almost like people call it a spheroid. It's a bit like an egg structure. So you've actually got like this shell of these blue cells with this bulk of these inner cell mass cells, the yellow and orange ones. But what I'm interested in is how do these ICM cells, so this bulk of cells here, how do they actually become either yellow or orange? So you'll notice, oh, you can't see my cursor, okay, I'll just wave a bit. You'll notice at this penultimate stage, we've kind of got this salt and pepper pattern. That's what they call it in developmental biology. So you've got a mixture of the orange and yellow cells. They don't specify in their final places. And so what I'm interested in is whether they're the microenvironment of those cells. So perhaps this orange cell here at the bottom, that's touching 5 cells in this simplified example. Does the fact that it's surrounded by 5 cells then kind of go on to influence that it does become an orange cell? If perhaps it was only touching 4 cells, would it have maybe become a yellow cell? I'm not saying this is deterministic, but I just want to start being able to look at this in a quantitative way, not just having a look at an image and saying, I think this happens. I want to give some numbers to what's actually going on. So my main research question is, how are the epiblast and primitive endoderm lineages specified, and then eventually how do they go on to organise themselves? And this can be broken down into 3-ish research aims, and I'll talk about 2 of them today. So the first one is, okay, we're talking about microenvironment. How do we classify, how do we quantify it? What's a good kind of way of describing that static cell neighbourhood? So I've got a picture of an embryo in 3D. How do I describe what's going on? And that's kind of like my first work package, as it were. The second thing that I want to do, and I'll flick back, I'll try not to do this too much, but I've just spoken about a period of time where that embryo is developing over the period of 4 or 5 days or so. So there's this kind of cell history that we've got going on here. And so what I want to be able to do is to link that historical spatiotemporal information of those cells with what they eventually become. And so in that sense, what proteins are they expressing, which can then let me know what kind of cell they're becoming. Are they becoming yellow or orange? And so in order to do this, I need to be able to link information across different imaging modalities or different types of imaging in order to understand this and make that link between historical cell information and what they become. So these are the 2 things that I'm going to be talking about today, and I should have said at the beginning, but I encourage you to ask questions throughout. So if there's something you don't quite understand, feel free to raise your hand. I may say it's coming up in another slide, but please do. So to start off with, I'll talk about some of the the approach that I developed in my PhD to quantify the cell microenvironment. And we're going to start off from the point that we have observed an embryo, we have taken an image of it, and we are able to extract the cell centres. So we can get the three-dimensional coordinates for every cell centre through segmentation approaches. That in itself is a research area that some of my colleagues were looking at. That's not trivial in 3 dimensions, but we're going to start from the point that we've got a list of the cell points in those 3 dimensions, and I'm going to then analyse these after imaging. So the way that I approach this, and there's not one way to go about this, but I developed a pipeline called IVAN, which is the internal versus external neighbourhood quantification pipeline. Let's just say in my PhD I wanted something flashy to talk about, and so I gave it this acronym. But what this allows us to do is take that list of 3-dimensional points, those cell centres, and then start to assess the environment and what's going on spatially. So I'm not depending on any biological knowledge, we've just got a list of points here. We can combine other information, but fundamentally it's built on this kind of like geometric analysis of the points. So to begin with, we were able to classify the outside cells. So if you remember, I spoke about it's almost being like this egg kind of structure. I use a convex hull to specify what those outside cells are. And obviously it's biology, so it doesn't always play fair. And so this is all set within a graphical user interface, so you're able to correct it when perhaps you can look at those cells it's classified as being outside, and you know from experience that that's really not an outside cell. Let's fix this. But on the whole, when we were testing this, by comparing it to images where we had a protein that would tell us for sure whether it was an outside cell or not, it performed really well. So I know the convex hull is perhaps nothing flashy, but it gave us a really good approximation of this structure within the mammalian embryo. What we could then do also with IVAN is to assess the cell neighbourhood or the microenvironment. And so again, using something really simple, using built-in functions within Python, we use the Delaunay triangulation. Something that I never got round to trying is various other ways of almost forming that network in between cells. There are different ways we could have constructed this, and it would have been interesting to see how that varied. But for the purpose of IVAN, it was a really good starting point. It was fairly simplistic, quick to run. And when we attempted to validate it by looking at images where we actually could visualize the cell membranes, it seemed to perform really well. I will caveat that with that's really hard to quantify the number of neighbours in a 3D mess of an image. So my validation statistics, is it much better? I tried really hard and it looked really good, and we had several embryologists who are far more familiar with these quantify these neighbourhoods, and it seemed to centre around what we could predict from the Delaunay triangulation. And then thirdly, within the base version of IVAN, we were able to use bespoke thresholding approaches, again very simplistic, but it would allow us to account for these more complex structures. So for instance, when we had the cavity, if we just naively use the Delaunay triangulation, what we'd end up with is, if just orient you guys, we've got the ICM down here, that cluster of cells, the cavity here, and what we'd end up with is this cell being a neighbour of this cell, for instance. And that was just non-physical. And so we just developed some ways that we could threshold this, but it was all optional. So it depended on your biological system that you were looking at, whether you wanted to apply that or not. And that appeared to be really effective. So that was a whistle-stop tour of IVAN. It was all set in a Python app, basically, and we introduced features through discussions with the biologists where we could not only analyse this spatially, but then we could start to include some metadata about the individual cells, so for instance protein expressions, and visualise that through the pipeline, because that may help you when correcting the outside cell classification, for instance. So using this, it was built to be easy to use. Sometimes it was a bit of a hassle with different versions of Python libraries, but we can work past that. But otherwise, I worked on this with colleagues who were developmental biologists through and through, and so they didn't want to get into the code, they didn't want to do that, they just wanted to be able to interact in a really flexible way. And I think Ivan achieved this, but it also allowed us to add new features really easily. We didn't make it too black box, and so if anyone was bored one day, you didn't have anything better to do, I'd encourage you, you could download the current version, break it, add in your own features fairly easily. I've not wrapped it up too much. But like I said, it was developed with biologists to work alongside their existing pipelines and be easy to use. And so I was really pleased with how this came about, and it allowed us to really start to look at what's happening within the embryos with these structures that we can qualitatively describe. How can we start to look at this in a different way? So that's the pipeline, and I just wanted to show one example that we could do with some nice images as well, because I like presenting this work. But one of the things that we do within embryology is we can culture embryos outside of their natural environment. So we can take an embryo at an early stage, and then whilst imaging the embryo, we are able to let it develop. So perhaps we would collect the embryo at 4-cell stage, and then we would allow it to develop until the 64-cell stage. And that's really cool, because then we can start to see what those cells are doing and how they're developing. But what they're growing in is really important. So at the top here, so these two pictures here, this is in kind of like an optimum media here. So you can see we've got that shell-like structure on the outside and this ball of cells on the inside with this empty space underneath. If, however, we had cultured that embryo in a slightly different media, which corresponds to this embryo here on the bottom, you can see we've no longer got that mass of cells on the inside. Instead, it very much looks like it's flattened against the outside cell layer. And so we know, it's a fairly historical experiment here, but clearly this embryo is not developing as it would naturally, and what we see when we collect embryos at that stage directly. And we can describe that qualitatively. I've got a flattened ICM, okay. But what we could start to do is actually then show that using the Ivan pipeline and actually give some numbers to it. What happened if we had an embryo that was kind of like suboptimally cultured and it wasn't as clear what that difference was? Well, we could use Ivan to help describe that and give some kind of different statistic, perhaps. So that was a whistle-stop tour of Ivan, and I promise I will hopefully slow down a little bit. But I was really excited. So this paper by a collaborator of ours, by Stanley Strawbridge, he developed his own method to account for inside and outside cells. And as part of his work, he kind of road tested Ivan. And I was really proud of the fact he didn't contact me once to say, how do I do this? It's broken. So it seems to be somewhat robust. But his work is really interesting. Again, just a slightly different approach to looking at this kind of spatial organisation of embryos, within the embryos, sorry. But as I've stressed throughout this quick description of Ivan, it's all really simple. Nothing that I've done there is particularly flashy. Perhaps I had a nice colour scheme within the user interface, but it shows us that within applied mathematics, I was able to get some really new and novel information from just applying these really simple algorithms and looking at the data in a different way. And I was actually able, although I've spoken only about the work with the embryos, I was also able to use Ivan on thin sheets of cells it was a kind of collaboration with a pharmaceutical company where they were looking at how the arrangement of those cells within these skin samples differed between different patient groups. And so it can be applied to different biological settings as well. So just to flag this here, and I'm not sure if the QR code ever works, but I can share these afterwards. But this work, along with a lot more testing and a lot more examples of what Ivan can do, I published back in 2021 now. But this was just like the first step within understanding how I can start to analyse these images of embryos that I was getting as part of my PhD. Okay. So moving on. Now, this is the bit that I want to spend the most time on, so that's why I've kind of rushed through, but hopefully it gave you a bit of context, is we've now got a way on how to quantify the microenvironment within the embryos, within a static image. But what I'd like to be able to do is say, culture those embryos, look at what the cells are doing, and then perform what we call an immunostaining, and I'll show you an example, and see what proteins they're expressing and link all of that information together. So if I show you an example, so this is our culturing of an embryo. So you saw the 2-cell stage. It's just gone into the 8-cell stage there. And these green blobs, this is because we've chosen a specific type of mouse so that we're able to track the cell centres. So what I propose to do in my later research is that we will track each of these cells whilst that embryo develops. And so you'd get kind of a map of what that cell's doing, what cells were its parent cells and what cells were its children's cells. And you can see just at the end of the movie there, before the embryo ran out of shot, because they like to do that, you can see we've got the formation of that cavity I keep talking about here. So you can see we've actually covered quite a wide span of time there. But once we've finished that, that culturing of that embryo, we can subject it to immunostaining. So here we look at different fluorescent proteins which attach to different proteins of interest. So the red cells here, we've chosen a protein that is indicative of the primitive endoderm fate. Orange cells, maybe, I think it was in my schematic. And then the white cells are associated with a protein that is associated with the epiblast cells. So we can find out what they become, what those cells became. And what we want to be able to do is say, okay, this cell in that final frame of the movie is this cell here. And then maybe see, okay, so that expressed that epiblast protein really strongly. And what did its neighbourhood change like during its development? Whereas, let's then contrast it to this cell here, where we've got this really bright primitive endoderm cell. Maybe that neighbourhood was slightly different. But I've shown you this matching. I would probably give myself a 10% chance of having actually got that right. It's really hard to do because in between these two images, we have to stop the embryo from developing any further, and so it often collapses ever so slightly. Me especially, when I'm staining the embryos, so the experimental procedure to do that, you ping them all over the place. They're rotating, you can't keep them in the same orientation. And so it's really challenging to match these individual cells, even with a landmark feature such as the cavity. Within that dense ball of cells, that can be really difficult to do. And so we want to develop an approach to do that. And that's what I'm going to talk about now. So this is an inverse problem, and we can set it up in the following way. So we've got two unlabeled point sets here. So the top one, so Y1 here, this is the cell centres from that final frame of that embryo culture image, embryo culture video. And then we can have the stained coordinate set here as Y2. And we want to be able to match these in some way. So there are a couple of points here. We want to be able to say this cell in the second image corresponds to this cell in the first image. And there are a couple of things. The point sets may not be the same length, so you'll notice I've got N1 and N2, and that could be, I think I cover this in the next slide, but it's important here. It could be due to cell death or cell division, or because we didn't segment the image particularly well. You'll see slightly, these images are quite fuzzy, and as you go through the embryo, that signal gets lighter and, less strong, and so it's harder and harder to segment, and so sometimes we miss some of these points. So you can have different numbers of cells, and also the first cell in Y2 does not necessarily correspond to the first cell in Y1. So we have no idea here. So what we want to do is develop this describe this transformation, which accounts for that rotation of the points during the staining and secondary imaging. Perhaps, this is optional, account for some kind of non-linear deformation. The reason why I say perhaps is the way that we formulated this, it's really intensive, and actually we kind of show that you don't necessarily need it at every point. And then we need to identify or include within our transformation the permutation. So what ordering of cells in Y2 is necessary in order to match with Y1. And so we wanted to formulate this in a Bayesian setting so that we get some kind of uncertainty on the cell matchings that we identify. and also the transformation that we observe, because you can imagine that in some cases there could be several transformations that would provide you equally good but a bit rubbish fits, and so we want to be able to assess that and look at how they compare to each other. So limitations of the data. I've already spoken about the difference in the cell numbers, and this makes it really hard. So if you've got 5 points here, 5 points here, great, we can find that matching. But if you've only got 4 points and 5 points, well, these 4 could match to these 4 equally well as to matching with these. You kind of have different subgroups of matchings. And so within this work, and this is what I'm super excited about, was we introduced something called Bayesian data selection. And I'll show you with a simplistic example here. We've got these points, we're just going to think about them in 2D for now, and hopefully you can see that Y2 is just a rotation of Y1. But we've got these 2 points here highlighted in pink, which I've just randomly moved them. They no longer truly match with each other. And so this is something we could observe within the embryo data set. And what we want to do is identify the transformation between these 2 point sets was sort of, I'm going to use a terrible term now, but like tempering out the contributions of those 2 spurious cells here. And we do this through data selection, so by inferring on the transformation, but also by assigning fidelity parameters to each of the points, the yellow points, which describe the contribution of that cell to the likelihood. So in this case, so for the green case here, our fidelity parameter would be much closer to 1, so it's contributing to our likelihood and our matching. We're paying attention to that cell. Whereas for this pink cell, which doesn't have a true match in Y2, what we want to do is infer on that fidelity parameter and see that this is going towards 0, so we're tempering out that contribution there. A slightly more abstract example to move away from that landmark registration kind of problem is, for instance, say we've got this XY data here, and between x equal to 0 and x equal to 1, we've got some kind of data that's linearly distributed, and thereafter it's exponentially distributed. Say we don't want to formulate a really complicated model. I say really complicated, but you get the gist. And I just want to fit a linear model to this data. If I was to do that naively, I'd get something like this red dashed line here. It's not really fitting any region of the data very well. If I fit this linear model and infer on both the linear gradient and the fidelity parameters on each of those data points, what we can see is that we infer values of the fidelity parameters in that linear region, which are much closer to 1, and then when we get to the exponential region of the data, you can see that those fidelity parameters are coming down to 0. So we're tempering out their contribution when fitting our model. So that's just a slightly more abstract way to consider it. Okay, so I'm going to go through the kind of setup of our problem here. It's going to be a little bit like we jump into it, but that's because I want to show you some of the results. So hopefully I can walk you through step by step. But we're going to, first of all, we define our observation operator, which I've shown on previous slides as being G here. And this is some combination of our affine transformation, which is the matrix A and the vector B here. We have the option to include the deformation. I'm not going to talk about that today, but that's this D term here. If we aren't including the deformation, this would just simply be Y2. And then we include within our observation operator our permutation matrix. So this is what's describing that matching of points between Y2 and Y1. We can then define our likelihood as a function of the mismatch between each observation and the observation operator for a given transformation parameter set and permutation matrix. And if we do that without data selection, you can see we've just got some generic likelihood function here, and then we define this is our mismatch, and we look at this for each point within Y1. So we're looking at the XYZ coordinates in Y1 of cell 1, and then within our observation operator, the first cell as defined by the permutation matrix after our transformation. And we just look at all of those mismatches. When we then include data selection, there's a slight modification here, so we still have our likelihood function, And we can see we start to incorporate our fidelity parameters here for each of the mismatches. So you can see if gamma 1 here was close to 1, then that mismatch would be contributing to our likelihood. If, however, it was tending towards lower values, then that contribution would be reduced within that likelihood function. And so effectively, each fidelity parameter is representing our belief that that cell in Y1 has a corresponding match in Y2. And then I'm skipping huge amounts here, but we can construct our posterior probability density just using Bayes' theorem, and I've omitted part of the normalization constant, which is dependent on the fidelity parameters that we have, but the same concept stands. So we have to define our priors on our transformation parameters as well as our fidelity parameters here. And for the affine parameters, these are very standard. They're just mean zero parameters, and we define their variance. If we are including the deformation, we use quite restrictive priors on the deformation parameters, because otherwise what can happen is we have really non-physical deformations of the embryos that we're just not going to see, and so we're just trying to make our job a little bit easier at this point. And then we also include beta priors on each of the fidelity parameters. So each fidelity parameter that we include, so the N1 fidelity parameters, we define our beta prior on that. And then again, something that I've glossed over but took us a huge amount of time, within our likelihood function we've got a noise covariance matrix, and we have no way of estimating this in a true setting, and so we included a conjugate prior on the noise covariance matrix, which was, we chose an inverse Wishart distribution. I'd be curious in drinks if anyone else has used it and had such a headache with it. But yes, so we just define our priors and we've got our posterior distribution here. So our posterior it's really quite complex. It's multimodal, so you can imagine you've got subsets of points that can match really, really well and get stuck within our matchings. We need to be able to explore that parameter space effectively. And it's also a mixture of continuous and discrete variables. That permutation matrix is inherently discrete. We're talking about matchings of discrete cells, as it were. And so the way that we formulated this was to use a random walk metropolis within Gibbs' approach. And so our approach was quite modular. So for fixed values of the permutation matrix and the fidelity values, we would then sample on the transformation parameters and so forth through this. And this allowed us to use adaptive MCMC, trying to, again, explore that parameter space well. And we also used tempering, because without this, especially in the complex cases, we would just fall into a well where we had maybe 10% of the cells matching really, really well, and then it was really difficult to get out of that region of the state space. So there was a lot going on at this point, fairly high dimensional as well, and we had to be careful. I'm going to skip through them. Had to be careful how we were making our proposals. We had angles within the affine transformation which were periodic. We also had bounded parameters, the fidelity parameters, so we needed to make sure we were proposing well on these. And also we had to think about how we would make proposals on the permutation vector. And we chose to make proposals which were one switch away. So we would just we would make the proposal of perhaps cell 2 and cell 3 would swap round and nothing else. We did play around with what happens if you made 2 switches, but we didn't see that much of an improvement. Okay, so that was the setup of our approach, and I'd like to show you some of the results now. And I'm going to start off with some in silico examples and then move to actually applying this to the real data. So to get you familiar with the kinds of figures I'm going to be showing today, what I'm showing you here in this matrix is the proportion of times at which that cell in Y1 was matched with this cell in Y2. So that top left corner is the number of times that we'd have cell 1 in Y1 matching with cell 1 in Y2. And you can see for this, it's a really easy problem. This is just us. We defined some points from an image of an embryo, and we applied a random transformation to it. And we didn't permutate the cells, but this doesn't make the problem easier. It just happens, it's nicer to look at, basically. But we can see from this really simple example, we're getting really good overlay of our Y1 cells. So these were the original cell positions that we had in purple. And then we would perform our MCMC, identify our maximum a posterior values of the transformation, and we could overlay these and see there's really good matching here. And then the final thing to unpick from these figures is this bar here, which are our fidelity parameters. And you can see they're all, maybe you can see, they're all shaded similar kind of grey colour. They're all around the value of 0.8. And you may say, well, why isn't it 1? And this was just an artefact of the beta prior that we chose on them. So this was actually the kind of expected value of the fidelity parameters for this setup. Okay, so this one, it's good to orient ourselves, but it's a little bit boring. So let's move on. Another in silico example here, but to show you what effect the data selection is actually happening. So we took a 33-cell embryo and we removed 3 cells at random from that data point in order to generate Y2 with a random transformation. When we didn't include data selection, so we're not including those fidelity parameters, all matches have the same weighting within the likelihood. This is the matching that we would obtain. So this is just a single example. You can see, perhaps on the whole, the embryo is sort of in the right orientation. But in this denser region, it becomes less clear how well those cells are actually matching. And we don't know how well they're corresponding with each other. And they're typically larger distances between the original points and the transformed points. If instead we include data selection, we end up with a matching that looks more like this. So you can see we're getting that really good alignment of those cells. And if we look at the permutation matrix, the permutation samples here, we can see we've got this offset diagonal by 3 cells, which is what we expected from our setup of the problem. And 3 cells in Y1 were associated with very low fidelity parameters. So those cells are being tuned out. So the data selection was having the effect that we were hoping for. And I think this is one of my favourite things. It just shows quite how powerful it was. I know it's a simple example, but we tried this in 64-cell stage embryos up with 6 cells difference. So we're looking at a 10% difference there, and it was behaving really well still. The next thing we could do, I was fortunate enough to work with a really talented postdoc. He was a surgeon and had incredible hands in the lab, and so was able to micro-inject individual cells within the embryos so we could tag them with the fluorescent protein. So these 4 cells in red, he would inject them and be able to make them glow like that. And what we could do is image the embryo after we've injected them, then effectively blow the embryo across the dish so that it would rotate, move around, image it again, and then try and match the cells in between these. And so this is a 16-cell stage embryo, and we could then match up, okay, so we know cell 22, 14, 5, and 13 were tagged, and we knew which ones were tagged in the corresponding image, and we could validate that. And we got really good agreement in those as well. So we're really pleased that this was working to this point. Okay. So I've shown you quite a few in silico tests there. So what about the real data? So our real data, like you've already seen, looks something like this. These are 4 embryos that we co-cultured. They're quite like growing together. And then we've got our stained image here on the right. We actually have an added challenge here that we don't know the embryo matching to begin with. We can make an educated guess. So for instance, embryo 2 here doesn't have a cavity, and embryos B and C in the stained image doesn't. But we don't know which one it is. And this is something that we can learn from our approach. So if we consider the 2 embryos with cavity, so embryo 1 and embryo 3, these 2 here, we can compare them with the 4 embryos in the stained image, and we can start looking at their permutation vectors here, at their permutation samples. And what we found, kind of skipping ahead here, but that embryo 1 with embryo A was a really good match. You can see that we really fell into this kind of optimum fitting here with a lot fewer cells with really reduced fidelity parameters. So the majority of cells have got a cell in both images, or they've got matches between the 2 point sets. And this is something we classified. We started looking at the average distances between all of the cells and their most likely matching and so forth, but there's quite a few ways we could have perhaps looked at describing this and assigning a probability of matching, for instance. So if we look at this in a bit more detail, so this embryo 1 with embryo A, we did notice that there were these two cells with really low fidelity parameters, close to 0, and we were a bit unsure. We were pretty sure of the embryo matching. But when we looked at this, it looks a bit of a mess, but trust me on this one. These points, so the pink crosses and then the solid pink ones, when the embryos were being segmented, so getting the cell centres, these cells were always in those low intensity regions of the images. So actually, I had just done a really bad job, and I should go back to the images and look there and see if I'd missed some of the cells. So it's a really valuable approach. It can actually help us improve our own segmentation. I could then rerun and see how things performed then. Okay. So this is a really complicated problem, and I was shocked when I joined the lab that this was done manually in the lab before. After 5 years of working in that lab, I still couldn't do it, but maybe I didn't try so hard because I wanted my approach to work. But it can, although I'm looking at this in the context of embryos, it means I can show great pictures, but it could be applied to any landmark matching problem. Perhaps they wouldn't be as complex in some ways, but the general principles would stand. And I think that the incorporation of data selection, in our case, was crucial for the successful application of the matching. But we often have imperfect data, and so it could be applied more widely as well. And this was published, this work, it's a mammoth of a paper. I was talking to Xander about this because we just wanted to show everything that we had, but I break down some of those modular approaches that we've got in a bit more detail there, and I'll be happy to talk about some of that following the session. Okay, and I've got one final section, I promise. It's like one slide or maybe two. I said Bayesian data selection was really the big finding of that work. The landmark matching application was really useful within the lab, and I got to do some really preliminary work with this. But the data selection could be applied in really different and diverse ways. And so within my postdoc, before my position now, I was looking at the mechanical modelling of tendons, and what we would have is data from experiments where effectively you would get a tendon sample, you would connect it to two clamps, and you would pull them, and you would record the stress-strain profile. Fairly fundamental. But what you don't see is during that experiment, some of the fibres are starting to break, and our mechanical model didn't account for that. And so you can start to see we're going to have deviation of our data from the model that we're applying, just simply because we haven't described it. It's too complex at this stage. And so what we did, we included data selection. So for every data point along here, we would introduce a fidelity parameter, and we would infer on the model parameters and the fidelity parameters. I spent a long time designing a prior for the fidelity parameters in this case. We could have used the beta prior, but it wouldn't make sense. So if I go back, it wouldn't make sense for a fidelity parameter to be 1 all the way up to here, randomly drop to a really low value. Then it would fit the model really well again. We need to consider the data as a progression and that correlation between them. And so I developed this prior, and off the recording, I always called it a squid prior. It looked like a squid with the legs. But anyway, my supervisor did not like that. But effectively, we knew in these early strain regions, it's really unlikely we would have damage occurring. And so we had really restricted prior in this region. But then we were less sure as the strain increased, and we encoded some of this correlation through the use of a logic Gaussian process prior. And we were really encouraged because what we started to see, so this is an example of one data set here. The data here is in yellow, And then our model fit is in black, and we can see at around this point we get the fidelity parameters in purple starting to decrease and then very much not contributing. And you can see that's where we actually get that deviation of our model fit. What's exciting, and I couldn't show here because that's a whole nother talk, is we had lots of data from different tendons, and we can see that damage occurring at different points for different data points that we would not be able to tell in any other way. Okay, it was a bit of a race through my research, but what my summary from this talk is, and it was really great to be looking back a bit more, is using simple quantitative approaches alongside more complex parts is great because you get to learn more about these really applied systems, and I think that's why I've really loved this research. But I also think that our use of Bayesian data selection could be easily applied in a variety of different ways, and I'm really excited to see how it's used outside of embryos or outside of tendons. I'm really looking forward to seeing other people using this approach. So with that, I'd like to say thank you all so much, and thank you to Katerina and Zander for inviting me. I've had a great time today, and I'd love to talk with anyone who's interested about any of the stuff that I've shown today. So thank you so much.